is 6. S as in San Francisco, yes. Q as in Quebec. And the number 5.
Okay, let's load the structure.
And the first question I would ask you is, the moment you have such a PDB ID from whatever, your research or your project partner, what are the first things you would checklist before then even diving into the first step of molecule design and whatnot?
So ultimately, what I would do first, I would actually look at the resolution. So let me quickly ramp up the RCSP, copy the PDB ID, and let's take a look at the structure.
If you don't like the resolution, we are not going to change the PDB.
Fair enough. So the resolution is 1.84 angstrom, reasonable, so very good. We are also all in this. The validation values are also seem to be pretty fine. Fairly recent structure, so 2019. Expressed in E. coli, that's cool. And it's from the Mycobacterium tuberculosis, as you have already said. Yeah, seems like a reasonable structure to work with. I wouldn't even go further to double check everything here, like the EDR map or electron density maps. So I'll just blindly trust the structure for the moment, for the sake of accelerating things a little bit. Okay, so what do we see? Before I actually pick the ligand to work with, do you actually want to explain the challenge to me?
The challenge is we have to, the ultimate challenge is we're going to design an inhibitor, a nice inhibitor, starting from this fragment. But in the process, we will address several questions. So challenges would be to, I'm going to list it now. So we're going to try to see if we can work with a different scaffold than what's in there, or explore different scaffolds. Then we're going to see, we're also going to find out if there are similar structures to this, which to the PDB, which could then potentially lead to selectivity issues. And if it is possible to address these selectivity issues, we're also going to see if it is possible to also design a covalent inhibitor. We're just going to see. If the answer is no, it's no. And the grand challenge would be, we would collect the list of our designed ligands and compare it against what is known. So the one ligand that came out of this exploration in results, and we will see how close we are, how further we are, and would also give us a chance to retrospectively analyze what we've done.
So since we don't have a script, I'll just randomly share my thoughts on this compound.
Yeah.
So what do we see here? The first thing you will see if you click on this compound, that our optimization, so basically we run an additional optimization step when we load the PDB to adjust the H-bond network for each structure. And you will see that we are still pretty nicely covering the initial starting point from the RMSD perspective. So no huge surprises here. Commenting on the structure itself, we have a phenyl ring system, a trifluoro-2 group here, likely forming lipophilic interactions because all the fluorines seem to be quite happy here. We have an unsaturated double bond here, so a vinyl group basically to the vinyl ring system, and we are ending with a carboxylic acid. So it looks very fragment-like-ish to me, so a very, very, very small molecule. It doesn't surprise me that all the calculated height scores for this compound are green, because this is a very obvious thing. So fragments usually form very high quality interactions within the binding site. This is why they bind. And this is kind of like reflected by the fact that every single atom here seems to be quite happy at where it is. So I will just click on this one and define the binding site by pressing the apply button. And we will see that now the side chains have popped up. I will now again zoom in a little bit on the structure itself. What do I see here? Just to talk a little bit about the interactions, maybe I will also switch on the binding site here. Visibility, surfaces. I have to adjust it here a little bit because it always interferes with the go-to interface.
Right.
So the binding site is there. I will recolor it to match the, again, go-to interface, God damn. It's so funny, it's actually a problem. So now the interface is colored, blue regions for hydrophilic and everything else for the lipophilic part. Let's not unclip it. Again, zoom out and toggle the transparency. Okay, this is good. So what do we see here? In total, I would say I was almost right, or I was right. We have a very lipophilic binding site for the 3-fluoromethyl group here. Then we see some hydrophilic areas in this region, which is also a little bit dominated by the phosphate groups of the co-complexed ligand here. Then what else do we see? A lot of methionines. Do we also see, I see a tyrosine here. So maybe this is also something that affects the pi-pi interactions with the phenylring system here. Besides that, a lot of lipophilic area. Yeah, so this is what I can comment on here. I will maybe turn off the visibility of the chain. Lina. Okay. Let's look at the interactions. So for seeing the H bonds, I will just transfer to the analyzer mode. And then I will see a very interesting arrangement. So basically we form hydrogen bonds with the tyrosine here, another interaction with the ribose moiety here in this area, and also coordination by this water molecule in this area. Okay. The next thing I will do is actually I will check about the size of this binding pocket. So when I define the binding site, all the residues around my compound are automatically included. Yet it doesn't mean that I don't have much space. That is a little bit, that the whole binding site is covered. So therefore, I will just double check with the binding site mode, check for unoccupied pockets. This is the binding site we are referring to. We see it's a little bit progressing further, so I will just add other residues. Ignore risks and proceed.
Just like we might have to calculate these cross one more time, not a problem.
Maybe I will switch on at least some water molecules. And everything. Molecules, waters. Oh. Ah. Again. Zip. Zip. So, okay. So again, next side only. We have to solve this somehow. Okay. So my proposal would be maybe I will start by improving the binding affinity of this compound and see if I can find something else. The first thing I would actually calculate the binding affinity once again. Let's also calculate the H-bond network, torsions, and clashes. All green, so seems like we have a very nice starting point in this area. Let's see. So
My first question would be,
yeah,
if I could interrupt. So you and I know that we have these cool tricks to generate compounds really fast. But if we could spend like 2 minutes by looking at the compound and the binding site, just looking at the surrounding from basic medicinal chemistry intuition, what would be the first kind of manual edits you would do if you have to look at this molecule with pen and paper, for example?
If I would look at the molecule at pen and paper, so what I would interest me, obviously, I would switch off the surface for a moment. We'll see in a moment why I'm doing this. So the first question would be, where is space to modify something? So what I will turn on right now is to actually turn on the unoccupied space feature, which shows me areas within the binding site not yet covered by the ligand itself. And you can kind of like see the very hint of some grayish area here. And this indicates that there is enough space to place a fluorine atom in this region. And if you have a lot of those grayish bubbles, it means there is a lot of space for a lot of fluorines. So ultimately, the question I will have now, where can I place something? So if I kind of like toggle it a little bit, you will see that there's a lot of space above my phenyl ring system here.
So as the cloud gets darker, there's like a lot more space that is there.
Yeah. So ultimately, it will be quite hard for me to grow orthogonal from the phenyl ring system. So I don't think I will fit something there. Yet what I'm seeing in this area is actually that there's a little room at this fluorine here. So maybe I can exchange it for something else and then use this one, maybe something lipophilic, maybe a theo something, or even a carbon, and try to grow into this area. Another valid idea is actually to see if I can work in the carboxylic acid region, because there is a little bit of space involved. You could try to put an ester there, could work, maybe not, or even an amide. I don't know. I guess there was also a water molecule here, so it will be maybe a little bit difficult to replace this one, even if it's highly coordinated. So in those cases, maybe if you recall correctly, there was a water molecule in this region, right, forming an interaction with this oxygen here. If I would have the time, I would also double check all related structures, if the water molecule is really conserved or not, to see if it's a conserved water molecule, it will be very hard to find something to displace it. If it's not that well conserved, I would actually try to find some substitutions there to match the H-bond network, like nitrogen, oxygen, whatsoever, and maybe utilize this. As of now, I will just assume that it's not very well conserved. But the right approach would be obviously to double check everything.
Should we try like 1 or 2 manual edits to see if we get into the right direction?
So, I said what I said. Let's maybe start with the fluorine here. So oxygen, and then I will be straightforward trying an toxic group here. See if this matches.
Should be able to see directly if we have clashes and all that, right?
I can, yeah, so torsions, clashes are not there, so there is enough room to actually post this toxic group there. But it seems like oxygen was a bad call. Which is like not
a fat corona.
Yeah, it seems like it doesn't. So it's not a price to be absolutely fair, because there are absolutely no valid H-bond interaction partners for the oxygen.
Yeah.
It was worth a try, you know, it was worth a try. Okay, let's try sulfur maybe. Sulfur is obviously bigger, so and it's also more lipophilic. Yeah.
Should we actually try generating a few conformers? Okay, this seems to be a beautiful edit.
So what you can nicely see here, the space was there. So it's a no-brainer. If you have a very lipophilic area, we have a methionine exactly in this position, highly lipophilic. We have this tryptophan facing the whole thing with the phenurine system here. I'm actually surprised that it worked out so well. Let's label it. So let's see if I've missed any interactions. So we have also this valine here, and also even the phenylalanine engages in lipophilic van der Waals interactions there. So I guess all in all, a good call from my side.
Okay, so essentially, we can, in a real world situation, we could imagine that chemists spend quite a bit of time here doing a lot of manual optimization, trying different kinds of edits, so to speak. And I'm voicing all of this out loud because, to be fair, I'm not a chemist myself, so I'm fish in a different pond. And I'm constantly trying to learn all of what happens in reality. So this is ideally what a normal chemist would do. And probably we can use some of these as starting points to explore the pocket a bit further.
Absolutely. So maybe I will just turn on the surface once again so we have a better feeling for the binding site and the complementarity here. Let's do like this.
Are you being notified if there are messages in the chat? Because I don't see any.
I have an eye on them, and so far no comments, but
okay.
People are free to comment. It's not like I'm advising against that. Okay, so actually, if you turn on the surface, we really see how nicely those fluorine atoms project towards the binding site. It's kind of like even, yeah, you have even this hump, and they really nicely align around the hump. Yeah. So maybe what I would try to do as well, I would maybe just try introducing a carbon just out of curiosity again. Maybe you can also start with this structure itself. This is, I think, what you refer to, and we do this design and do some template-based docking just to see some confirmations of this edit.
Yeah.
So you now receive a total of 10 poses. And yeah, unsurprisingly, also the decoration with the ethyl group now works pretty nicely. We even reach very nice LLE values here. What one could think about is actually, if I now use this one. So if this one fits, I would ask myself if maybe something cyclic can do the trick as well. So I would just close a cyclopropyl here and do some template-based docking. And yeah, I'm actually surprised. So it works to some degree.
At least nothing with absolutely horrendous clashes and whatnot.
No, it's more or less fine. So even recently, there was a post on Drug Hunter. I think we all know Drug Hunter. And they showed one compound. I was actually quite surprised to see that. It was something like this. It was a spiral system. And covered with one additional methylene, and I already completely forgot that it exists.
Are you talking about this so-called bow tie compound?
Yeah, I don't recall, honestly. The thing is, like, I'm always
Okay.
focused on the structure.
Right, because they actually essentially called it the bow tie compound because it had this sort of a motive.
Yeah. So let's do another looking. We have a question. The question is if there's a strategy to improve the LE properties of a molecule. So basically, how do you improve this one? And the only solution to this is actually to improve the estimated affinity. So our software calculates or estimates the delta G values, so the estimated binding affinity of a compound. And the farther you actually push it towards higher values, this improves the LLE, the overall LLE. So it's not like you can, it's kind of like a consequence that the LLE improves based on the affinity and the types of heavy atoms that you produce. So there is no perfect strategy to solve this one. Oh, this works pretty
To be
nicely.
fair, the LV that we display is also based on the affinity calculation algorithm that's HIDE.
Exactly. So in our case, LLE is just a basic result of our height estimation. Okay, but you see, this strategy could also work. To be fair, to be very fair, this cyclopropyl methylene group, it's highly prone to metabolism, I would say. So this is the first entry point for metabolism. It would be oxidized. So although the affinity improves, I highly doubt that this would actually be something that you would like to see in a final drug-like compound because of the metabolism issues coupled to that.
Could we also, as we do it, because you mentioned metabolism, could we keep a track of some metric that accounts for the metabolism as we keep designing these molecules?
Yeah.
Like we probably have one of our Optibrium properties that accounts for the estimate of.
So, like, let's look at the not paraglycoprotein. I would actually check for the CYP enzyme. So basically those two values. And this is exactly what I thought. You will actually have a high affinity to CYP enzymes. The medium is okay, surprising, but it's not low. So this would be the metabolism entry point for CYP enzymes. As for the other ones, values are okayish, not great, but not too high. But for 2D6, for the cytochrome 2D6, this is something we should keep in mind. And as you can see also, same thing. So every time you have a very long chain, those compounds are very prone to be metabolized by CYP enzymes. It's not a perfect solution to just introduce ethyl chains around your molecule just to improve the affinity because it won't do much by the end of the day in real life.
I would have done this and been happy, but now I know.
So basically the question is, how can we compare the Caesar estimated affinity with other docking tools such as Autodoc Diana? Ultimately, those are very crude informations. They are not empirical. They're based on very fundamental chemistry. They compare average compared to other softwares as well. So ultimately, there's a publication on that, the final height publication. It's okay. I have to admit, it's not perfect. No docking algorithm is perfect, again, to be fair. But you get a pretty decent idea of what range of a compound you can actually expect. So basically, we provide a range. The value we calculate is always in the middle of this one, of this logarithmic scale. And we add 2 orders of magnitude, so factor of 100 for the lower and upper range here. So if you really reach this picomolar section, you get a very good idea that your compound may be somewhat in the nanomolar activity range. So I always would my gut feeling is always to trust the upper value, just to be absolutely sure. But I've seen some examples where actually the lower limit was reached. So the range is quite forgiving, but it's actually worth it. So for an assessment, it's fine, but it does not replace any experimental data that you can collect on that. Okay.
Absolutely. And that's the phase of discovery where we are at. We're not gonna buy the molecule yet, but I want to shift gears and actually go and see if we can generate a bunch of new ideas based on one or two starting points of your choice. So what we're trying to do is explore the binding site better, explore probably a diverse set of chemotypes, newer scaffolds, and see what we can achieve in the next 10 to 15 minutes before we also find out if some of these compounds are within the purchasable domain that we can explore from chemical spaces.
So another thing that people can actually consider, and I really love the strategy actually, is to close a ring system. So right now I'm just leaving this one behind me and focus a little bit more on the right side to me. So what we see here, this ligand is actually quite nicely positioned into this area, but you can also imagine that it could be flipped also. And if we close the ring system here, introducing 2 additional atoms and do it like this, so a naphthalene here, I would like to check if we can kind of like fix the carboxylic acid in a particular position, which decreases the degrees of freedom and can greatly improve the affinity. So let's export this one. So let's see what the problem may be. And the problem will be very likely that we have clash. Well, I tried. I tried.
I mean, this is essentially what the tool is wanting you to do. Try different things, find out how poor your choices have been.
So I will just remove the clashes now. Let's go back to original compound. I will do maybe a smaller ring size. I don't think that will matter.
Collision of these red arrows actually affects you psychologically. So many of those reds.
So I will try something smaller. Now let's start with an oxygen here. I don't think that so there is a possibility that I find something here. I don't think so. But actually, see, if I decrease, if I make it a little bit smaller, so the 5-member ring system, I can actually quite nicely introduce a full-on ring system. Let's make it even sexier by thiophene. No, this is clash again. But it seems like the uran ring system is working okay-ishly. We do get some clashes, but it's possible to introduce it. Again, the question would be in this area, would be there an additional induced fit happening that tries to fit the full ring system? So it's methionine can flip around a little bit. So generally, it's not absolutely stupid to double check this compound. Because it's also.
No, we actually ended up with a newer scaffold now. So from a monocyclic system to a bicyclic one.
Exactly.
Perhaps now we have a chance to pull out all of the cards from our pack.
Do it. So, should I go with the one ring system or should I stick with the original one or the full?
You are the.
I am the one. Okay.
You are the one who's controlling.
So let's do 2 things here. I will now transfer my compound to the inspirator mode. I would like to do now 2 things. One thing is I would like to see if the program, if Caesar is smarter than me, I will try to find replacements for the tree fluoro group here, just to check what it will come up with. Let's go up to 30 compounds. I will use Inspirator. I will use my, not the hinge binder library. I will use this one, sp3 growing. I will use my
As you're trying to
100.
do that, I have a question from a very basic point of view.
Mhm.
Don't stop what you're doing, but why are you trying to replace this fluoro component that apparently would give you a lot of affinity based on simply being hydrophobic?
For the best reason, out of curiosity.
Okay, right. That's valid.
So ultimately, I liked the TO ether compounds that we came up with. Yet TO ethers are not so prominent, not so well established. Let's face it like this, especially with the two two fluoro groups. It will be kind of like not so easy to synthesize by the end of the day. Also, the purchasability of the building blocks would be not easy. So therefore, I would like to see if there's something, a little bit more accessible.
Right. That's fully valid because I guess a lot of new molecules are discovered solely from the chemist's curiosity. So
Exactly. And especially since we're now in the fragment stage where we just grow the compound, this is where chemists like to have. It's very straightforward. So very easy to synthesize, not so expensive building blocks. Therefore, I would like to see if there's something kind of like matching the vibe. So actually,
quick
you
question.
see,
Which
yeah.
library did you use? Meaning how many thousands of fragments were scanned in this time?
So I'm not using the default one. If you download Caesar, you have the 12,000 library. I don't have to tell you, but I can tell at least the viewers.
Right.
The default 12,000 I included of growing fragments. The one I'm using is the larger one, so 120,000, also available on our website as download. It's just the download package of Caesar would be too large if we include it as well. So therefore, it's kind of like if you want to have it, go ahead, but it's kind of like just one additional download. Okay, so actually, I'm a little bit, I wouldn't say offended, but it came up with the same strategy with the tire ether. So the thing is, the space is very limited, so I didn't expect too many. I don't expect at least too many. I would never imagine that this would fit there. It's quite surprising, but you also see the affinity is awful. Yet I'm surprised about the space though. But this is quite interesting because if we know that this kind of like matches there and the space for a 5-member drink system is there with even decorations like this hydroxy group here, one could reconsider if you can introduce something lipophilic there, like a thio, like a sulfur atom as a linker, and then attach the 5-member drink system to it. I wouldn't say it's a stupid idea, but it can be one idea at least. You actually have a lot of space there. So this is something that is comparatively synthesizable. I would also agree that I would assume that this building block is purchasable.
This is still within the fragment-like domain, isn't it? In terms of just looking at the size and descriptor that you think.
Let's take a look at the molecular weight. And the molecular weight is at 323. So the one above, which one I liked, this one, we are absolutely in the fragment domain. So 220, that's nothing. So this one is exceeding, yet I am, look at this. Is it projecting? Where is it going? So there's actually a lot of space to occupy. Let's not click this side anything. we can actually even grow into this whole area. So there is actually a lot of room. I'm quite surprised. Never expected that. So again, this is exactly what I was talking about, 5-membered ring system. In this case, even it's also possible to introduce some things that are very nicely contributing to the overall binding affinity. Yeah, so all in all, so this is exactly what I'm talking about. Introducing an additional linker. In this case, it's oxygen. I would suggest if you would like to have the linker also contribute to the binding affinity, so it's sulfur here. And the 5-membered ring system seems to fit there at least somewhat-ish. There's a lot of space. Clashes though, here and there, but there will be also better solutions. Okay, so all in all, a very nice growing extension here. What's the best one? The best one is surprisingly this little fella, but it's a little too exotic for my personal taste. But I like this one very much. I just mark it as a favorite. Okay. So we've explored one side. Now, out of curiosity, let's explore the other one. I will now exchange this carboxylic acid part here. So the last 20. Yeah, numbering. I have to come up with an idea how to differentiate them. So at least let's look at the S affinity maybe. This is the issue once you actually, let's delete everything except my favorite and the original one. Just rerun this whole thing.
You just have two molecules. Maybe show it in the grid view, and we see both better.
Yeah. You mean this one?
Yeah. Okay. Now we have way too many molecules.
Okay, that's something good. So those are now the results for this part. And to be fair, there is room to grow, actually. You can see how you can actually just kind of like sneak around and project towards this binding area here, close to the phosphate groups. Yet, I think it's quite difficult to find something. So all of them are scoring quite lousily, where I would say that any of those are valid ideas.
Should we also use your other idea from the editor where you designed a bicyclic system?
Yes, let's clean.
For some reason, that molecule was really nice, at least the way it looked.
Where is my bicycle? My bicycle. There's my bicycle. So maybe before we do that, I would like maybe to see if I find something without clashes. So let's do a quick docking run.
Ah, you're selecting a better starting point.
Yeah.
Okay.
Meanwhile, there's a question, is protein residue flexibility considered for fragment growing and binding affinity estimation? Two things on that. First thing, no, we don't consider any flexibility within the binding site in the scoring system. Everything is rigid. So we keep the side chains rigid in those areas. And so there's a little twist to that. Okay, for the initial points, I don't find anything better. So I'll stick with this residue. So we don't take protein flexibility into consideration, but we do take into consideration tautomers and flipping of particular side chains. So asparagine, glutamine, histidine, all of those side chains are actually flipping based on the ligand edits, just to see if anything would better match according to the hydrogen bond network. So we calculate in each single iteration, when we calculate right, we calculate the H bond network and the positions of hydrogens, and then adjust everything accordingly. So it's our protonation algorithm, which is called POTOS. But for the real flipping, so movement of heavy atoms, we don't do that in those calculations. Okay, bicyclic ring system. What would you like to have with this? Or what would you like to do?
I'm thinking, should we do a template docking to see if this can snuggle a bit differently?
No? Let's clean the generated process. And then do a template docking. So I pick this one to set it as a template, and then run my template docking. Yeah, some snuggles are there, but it seems like we can get rid of them too much. So orange is the best I can do for you, which is also, no, it's a little bit better. But this one seems to be the best one. Okay, what would you like to do with this scaffold?
Let's use this as our starting point and explore all directions.
Okay, so basically what I wanted to achieve with this one is to fix this carboxylic acid in this position. Two reasons for that, because this carboxylic acid is deprotonated, it also undergoes mesomery effects, and therefore it is always, or should be, very happy if it's in the same plane as the bicyclic ring system. This is what's my main idea, to actually introduce this ring system there. Ultimately, if my carboxylic acid is in this position, what I would like maybe to achieve is actually to have it in the same position. So I now use my core replacement, so recore. I will keep my G-fluoromethyl group here and keep the carboxylic acid and see if I can find something in this area. So based on the mesomeric effect here, I would like to actually have something that is also aromatic here. So I don't want any aliphatic atoms in this area, or at least I would like it to have some aromatic ones. Make the thermophore constraint a little bit higher, and let's go with something aromatic. What do you think of that?
That should essentially preserve the properties of the molecule.
Which ones do you mean?
We're trying to still set an aromatic constraint in the middle, right?
Yes, this is an aromatic constraint.
Yeah, so let's go.
So basically, we are now looking for 3D matching things for this area. So no far, this one seems good. I will just ask for a little bit more because I'm very greedy. And I will ask for another set. So it's now 90 solutions. Huh. So let's check. Okay. We haven't found the perfect match, but yeah, it's the same thing with the way around. This is hilarious. So in Indo, I'm actually surprised. Yeah, see, I will just turn off the microphone constraint so we can see things better. Now also, let me calculate the clashes here. Maybe they will find something without a problem. Clashes and Protheon. So a little clash, but I'm quite surprised that there is no clash for the nitrogen, to be honest. I expected, because look at this. Clash, but no clash. Because of the electrons. Then, oof, a lot of things happening here, but
yeah,
even
it's quite busy.
It's quite busy. Can I find anything without clashes? I mean, I'm not feeling it though. So basically, what is the takeaway here? This is also quite interesting. Even though this result is basically useless, there is one special information that is important to me, namely that there is also possibility to introduce a nitrogen in this area. So it's not only carbons, but it's also possible to introduce a nitrogen in this area.
Mhm.
And the question is, what's the force field used for the proteins in small molecules? Ultimately, it's a height-related force field. It's called geoheight. It's based on hydration and desolvation of the target structure and the ligand itself. Also, again, published the original height publication. So if you go to our website, CISA, and under science, there is a section about height, and all the original publications are listed in the PDF file there. So you can easily look it up. What else do we see here? So again, okay, this is a clash. I was actually quite happy to see that you can introduce something here as well, but the fluorine is too large. I think you could actually work with a meta group even in this area. So fluorine is a little bit larger. But even kind of like going through those examples provides with ideas how to actually complement things you could introduce into your compounds.
So
Okay.
let's say the assignment now is in this round of in your hypothetical discovery team, this round you ought to design like at least 5 drug-like or lead-like molecules to then take and see if you can purchase them. Ideally, I'd try to move them and use one of our combined filters to make sure that they at least theoretically fall into the drug-like and lead-like region. How would we go about it?
So maybe what I would like to do before we do that, I would take a bicyclic compound because I really want to find a better solution. I would take again once again to the inspirator mode. What I will do now, I will try an analogging run. So basically, this will just decorate your compounds, check for replacements here and there, just to get a feeling of doing things. So I'll go with 50 compounds. And I will just take the best scoring one to see if there's something commercially available.
Basically, you're doing a little bit of SAR with analogs, right?
Exactly. So just to provide you an idea, so 24 modifications are allowed. For the rest, there is not enough space, or a score would be too bad. And this is how it would look like. So basically, again, get an idea of where is space for replacement, or what can be modified because it applies common medicinal chemistry reactions. Common medicinal chemistry reactions usually don't involve too much of replacements of fluorines. So therefore, I'm not surprised that there is not much modifications in this area. We actually see now and then that there's also nitrogen somewhere, but we see also a lot of proposals to look actually into this direction, right? So this is what I like very much as well. So okay, let's go with this one. This is the high scoring one. I promise I will take the high scoring one.
Okay,
Now,
that's
copy this one as Mars.
easier to follow. We're picking the high scoring one.
Let's load it into Infinity. I would just use the chemical spaces because they have the most compounds and I'm more likely to find something matching my vibe. So the question would be which mode I would like to use. For those kind of cases where the molecule itself is very, very small, I'm not too much of a fan for using the analog hunter because it just provides you with a lot of ideas if the compound is already somewhat evolved, just to explore the decorations around the compound a little bit better and more efficient because it's analogs. I'm not looking for the analogs, I'm looking for the closest thing that I can buy of this one. So I would actually recommend to go with a motive matcher in this regard. I will load my molecule. And I will go with Galaxy because time is running out. And I will do an MCS search. So maximum common substance. So to be fair, this arrangement is not very popular. We are a two-row mature group. It's not always present in building blocks, I have to admit. We get an indole-related compound. It's an amide. So indole would be kind of like the closest thing we can get here. But maybe, you know what? Let's try it. Why not? Let's use the 5 compounds here just to see if there's anything that's at least somewhat interesting. Let's copy all of them as smiles. Go back to Caesar, transfer this one to the docking mode, get rid of this one, clean stuff up here, and now load our F molecules. 1, 2, 3, 4, 5. And again, let's do a template-based docking to make things a little bit faster. So this is our original. Let's maybe color its original pose red. So yeah, but you see, the amide is not well tolerated, at least in this regard. So unfortunately for the fast one, not so many nice surprises there. But honestly, we got some pretty nice ideas how to actually grow this fragment. So therefore,
Right.
I would say I'm fine with how far we've come within this hour.
Sounds good. Let's do just one little exercise at the end of it. Let's compare what these guys have done with the final compound that they ended up designing, and that is 5SQL.
L, like line?
Correct. You're right. Exactly. That's the compound.
5SQL.
Yeah, I'm wrong. It's 6SQL.
You're lying.
That's the one.
Okay, well, I'm actually I'm surprised that there's a conclusion to this one. It's actually one of this drug fragments and drug discovery reviews. Not that you mention it.
Yes, it is. It's one of those cases.
Okay. So,
but one of the important things that I want to look at is to see how much the binding site has essentially changed between our first protein and the second. So this gives us somewhat of a feeling for what the induced fit has done, or what the conformational change that the newer molecule has produced. Because to me, it also seems that they've gotten rid of the trifluoro affinity anchor themselves.
I'll quickly just move the chain. Okay, so I will load this compound into the analyzer. Really close. Okay.
Now you know why I was pushing you towards using this bicyclic system.
This is the original one, perfect. So let's pick the original one, make it red. Let's remove everything. Okay, so now it's a little bit cleaner. In the conclusion. So we actually do see that there's flipping involved. Actually, if you recall, I actually docked the bicyclic compound, and the best scoring pose was flipped by 90 degrees, as in this case.
Exactly. Yeah.
So I'm surprised that the docking was correct.
Yeah.
Nice. Okay, so what do we see here? So they came up with the same basic idea to introduce a bicyclic compound here. They actually target another binding pocket, so they project more towards the, this was the unoccupied space, right, that we saw above the ring system. If you think about that, it's just kind of like this would be the same thing of the, so it's just flipped a little bit. So this one, this mature group, moved in here and was exchanged for chlorine. So I don't think they actually saw it coming when they designed this compound. So this clipping was
Yeah,
actually.
I think so too. It would have been a very long and winding story to come from this fragment to this molecule.
Yeah. So then what did they do? They did do an bioisosteric replacement. So the carboxylic acid was replaced for sulfonamide. Sulfonamides, I think I would say it's fairly common to use them as a bioisosteric replacement. The idea is that the oxygens in this case are very similar. The distance is not that much, but the analog concept behind this one, 1.5 angstrom for both almost. But sulfonamides are far more favorable when it comes down to ADME properties. So carboxylic acids are just flushed away through the body, and sulfonamides are a little bit more forgiving. Then I would say this is a very uncreative solution. They have a phenurine system, fine, I don't mind. But what they're doing here is they simply introduce an amino group that likely will interact with the phosphate groups in this area. So cationic and negatively charged phosphate groups. Not very creative, but I think it addresses the task quite nicely.
It works.
So all in all, it was actually a fun exercise. I like
it.
it.
I mean, honestly, within an hour, we've spoken about different ideas, different strategies on how you can start with, and that is the whole point of What the Drug.
Absolutely. Absolutely. Before we finish, there is one more question. Height is Caesar is measuring the binding affinity based on the height algorithm. How much of an influence is the concentration of the compound is the binding affinity of this algorithm? Yeah, so basically this is a very detailed question about the height algorithm. I will follow up on that. It would be a little bit too detailed to explain right now. I'll just, again, the easy solution is to read the publication, but I will answer your question subsequently afterwards. So, your last chance to ask any questions. Meanwhile,
very good. Actually, thank you for having me.
I have to thank you for such
I can challenge you one more time sometime in the future.
Hopefully so as well. So thanks everybody
I
for
look
tuning
forward to challenging you with newer features in the tool, and then it will be a bigger challenge.
Looking forward to it. So,
alrighty.
Thank you. Meanwhile, for the rest of the Schützenfest, as we say in German, if you have any comments, feel free to text us, reach out to us, contact@biosolveit.com. Thank you for the nice comments. I hope you enjoyed it as much as I do. I hope you would like us to continue doing What the Drug